Welcome to Transmed's breast cancer analysis of the correlation between gene expression and HER2 IHC levels. By integrating clinical and molecular data, it becomes possible to identify correlations between molecular characteristics, such as variants, and clinical characteristics. The ultimate goal of this system would be to find variants or gene expression patterns that correlated with differential response to treatment as part of a personalized medicine research program. The public TCGA dataset is limited with respect to outcomes and treatment information, so the examples presented here will provide examples of the types of methods that might be applied to reach this type of research without necessarily identifying significant results in this public data. To start this exploration, we will log into Transmed BIS with TCGA cancer data. To do so, open a web browser and go to the URL provided with your BIS-free login info. Once loaded, you should see a login screen like this. Log into BIS with the username and password provided, and select the checkbox to agree to terms of use for BIS. Once logged in, you will see the Project Explorer home screen. Logging into the system controls what data you have access to, including whether you have identified or de-identified access to a particular project. Having user identities in the system also allows control of user artifacts, such as cohorts, reports, and analysis results. These can be created, saved, and shared between the users of Transmed on the basis of user identity and role. What you are looking at is the Favorites view. A workspace is where you will be doing all of your work. The workspace that was created for you to work in should be visible here. Specifically, a workspace with your login name as part of its name. We have made it a favorite already. To view all the workspaces available to you, select the drop-down called Favorite Workspaces and select the option called All Workspaces. Locate your workspace and double-click on it. This will open the Workspace Explorer tab. At this point, you will see the contents of the workspace. In the workspace are some pre-populated artifacts that are the results of running this scenario. We will not be using these here. We will create them from scratch, but have made them available so you can look at them as a reference. Workspaces are the folders that contain all user artifacts that are created by a user. Artifacts are organized into workspaces such that users working on the same analysis project can share items within the workspace and collaborate together on the project. This scenario utilizes Cohort Explorer, so we will need to open the Explorer. In the Tools drop-down, select Cohort Explorer. This opens the Cohort Explorer tab and shows you the unfiltered cohort of all available patients in the right portion of the window and the available data, or traits, in the left. The traits are organized into folders just like Windows files in the directory structure. You can at this point double-click on those folders and explore around. There is a back button and an up button below the Tools drop-down. Use these to return to your previous position. As you drill down into folders, there is a graphic representation below the back button, showing you your location in the hierarchy, with the highest level being All Items. These are hot buttons as well, so you can return to the highest level by clicking on the All Items button. To begin this scenario, be sure you are at the All Items level. Our first task will be to limit our cohort to only those participants who have breast cancer. This is done by limiting the participants to those that have a tumor tissue site of breast cancer. To do this, we could navigate to the Participant Traits folder, then the Participant folder, and find the trait called Tumor Tissue Site. This works if we know where to go. However, let's assume that we do not know where to look. So the alternative way to find it is to use the search function. Reducing the current cohort of all participants to those that have tumor tissue type breast cancer is easily done by using the search function. In the search box, type tumor and the return key, or press the magnifying glass icon. The results of the search are seen in the window below the search box. Find the trait called tumor tissue site and select it. Notice that when you select it, a few icons show up. One looks like a funnel to filter the cohort, and one that looks like a group of people to group the current cohort. The funnel allows you to filter the cohort and reduce the cohort to only those that match the selection of tumor tissue site breast. The grouping option takes the existing cohort and splits the cohort into groups based on your selection. We then select Filter on Specific Choices and select Breast. In this demo, there is only one choice. However, when other TCGA data is loaded, additional choices like lung will be possible. Selecting the Apply Filter button, or executing this filter, will take the existing cohort of participants and reduce it to those with tumor tissue site of breast. HER2, ERB2, is a key oncogene in cancer. Amplification of HER2 has been associated with a particularly aggressive subtype of breast cancer, but it is also indicative of response to treatment with Herceptin, trastuzumab. This example will show a method to determine if the expression of the ERB2 gene is correlated with the level of the amplification of HER2, as measured by immunohistochemistry. Now we're going to look for those participants with the HER2 immunohistochemistry levels. In the search box, type HER2 and type return or press the magnifying glass icon. When the search is complete, you can see the discovery of two traits where HER2 immunohistochemistry level results are found. One is the participant traits in the participant folder, and the other in the clinical follow-up V15 folder. For our purposes today, we'll be using the one in the participant folder. We are using this one because this is the unit of measurement that was taken nearest to the time that the sample was taken for measurement of the gene expression, as opposed to samples collected for follow-up. Selecting the result associated with the participant folder causes a filter icon to appear. This opens the Filter Options dialog. Click on the filter icon. Once the filter options are open, notice there is a choice of filtering on all the choices or filtering on specific choices. If we wanted to reduce the cohort to only those with 3+, we could select Filter on Specific Choices and select 3+ choice. For our purposes, we will filter on all choices. Selecting the Apply Filter button or executing this filter will take the existing cohort of patients and reduce it to those with HER2 immunohistochemistry level result. At this point, we're going to generate a basic report of these participants. To do so, we simply click on the Create Report button. This launches Cohort Reporter and pre-populates it with a basic template. Now we want to investigate the relationship of different drug use to HER2 immunohistochemistry level results. To do this, we need to add drug name to the report. Using the search box, type in the word drug and press return or the magnifying glass. In the results, find drug name and select it. Notice it is part of the clinical drug folder, which is part of the participant traits folder. Add it to the report by pressing the plus sign to bring up the options dialog. Because our interest is in the participants who have a drug use, we will use the constraint to limit the result to those who have had a drug assigned. Click on the constraint drop-down and select all observations. This will eliminate any participants that do not have a drug assigned. Click on the OK button to add it to the report. Now we are ready to generate the report. This is done by clicking on the Save and Close button, located in the upper left, right below the BIS 5.1 icon. This will bring up the Save New Report dialog box. Give the report a name, something like Drug Report HER2. Once you've entered the new name, click the OK button. This will bring up a dialog asking if you want to generate the data now. Select OK. Following the generation of the data, the report will load. In the report, you can see the data. Along the top of the report, you'll see the title of the column, and each row represents an entry. Notice that the columns are active and can be sorted similar to Microsoft Excel. After reviewing the trait values, let's create a pivot table to look a little deeper into the drug assignment and how they relate to the HER2. To create the analysis, we need to open the Report Tools by clicking in the Navigator window on the Report Tools icon. This will display the various reports that are possible in our base product. Select the pivot table tool icon. We need to add the variables, so select the Add Variables button. For values, click on the Values box, select Participant, and click on the arrow to add participant. For column labels, select the Column Labels box, select HER2 immunohistochemistry level result, and click on the arrow to add it. For row labels, select the Row Labels box, select Drug Name, and click on the arrow to add it. Click on the Done button. The table is now populated. To do some discovery, let's sort the 3+ column to have the highest drug counts at the top. Right-click on the header of the 3+ column and click on the Sort option. This sorts the result from lowest to highest. Click on the Sort icon to reverse the sort order. As expected, because HER2 amplified patients are more likely to respond to Herceptin, Herceptin is the most commonly administered drug. If we select all the drugs that have a result greater than or equal to 8, we can create a chart for these. Once the cells are selected, click on the Show Chart button. This displays a charting window below the table. We need to select the chart type. In the Chart Type drop-down, select Bar Side-by-Side Series 2D. This creates a chart of the selected results that is easily exported for reports and papers. Now select Hide Chart to return to the original view. If we wanted to have the percent of the participants for each drug, we can easily do that. Select Add Variables again, select the Values box, and add Participant again. So now there are 2 listed. Right-click on the second participant and select Field Settings. In the Edit Pivot Field dialog box, select Summary Display Type. Select % of row and press OK. Select Done on the Add Variables window. You will now see an additional column that shows the % of the participants that were treated with this drug. This is helpful in showing which patients are being correctly treated with Herceptin, those with high levels of HER2 and those who should likely not receive HER2, those with low or non-existent levels of HER2. Let's save and close this pivot table. Select the Save and Close button. Now let's look at the gene expression levels. In the TCGA data, we need to look for ERB2 expression levels. Let's go back to the report setup by selecting the Report Setup button. Because we saved this report, we will just modify it and save it again as a new report once we have what we were looking for. First, we need to delete the drug name trait from the report. To do this, select the clinical drug level, right-click on it, and select Delete Level. Confirm that we want to delete the entire level. Now the report setup is as it was when we started to work on it. In the search box on the left, type ERB2. Find ERB2 in the search results and select it. Notice that there are 3 possible results: gene symbol genetic variant, gene symbol variant observation, and the one we want, gene symbol expression. Select gene symbol expressions plus sign to add it to the report. This opens the Add Trait dialog window. Notice ERB2 is already selected, so we can select OK to add the trait. Notice that BIS automatically figured out the additional traits needed to connect participant to the gene expression observation. We will now add the trait gene expression. To do this, right-click on the gene expression observation level and choose Edit Level. This opens the Edit Level dialog window. Select the trait gene expression to add it to the report and click OK. Now, since we want this to be a new report, preserving our previous report for the pivot table, we will select Save As and give the report a new name, HER2 ERB2 Expression Report. Once the report is saved, close the report setup by selecting Close to return to the General Information page. Select Generate Data to run the report. With the data generated, you can scroll up and down to see all the results. Let's look at the data in a different way. Select the Report Tools view on the left. This shows us some of the different reports possible, including the pivot table we just finished using. This time let's choose box and whisker. For variable 1, choose gene expression. For variable 2, choose HER2 immunohistochemistry level result. Press execute. This produces a box whisker chart which shows that the levels of the expression of the gene product ERB2 seems to strongly correlate with the amplification shown in the immunohistochemistry. The 3+ patients have a much higher level of expression than the other 3 groups. Interestingly, there appears to be little to no difference in the other groups. Save and close the box whisker report. Let's do a similar exercise, but using BIS's R analysis tools. Select the analysis tools option on the left. This displays the basic R functions available in BIS. Users can add their own R functions to include in any additional capabilities desired. Select ANOVA one-way axis control. For the dependent variable, select gene expression from the drop-down. For the grouping variable, choose ER2 immunohistochemistry level result from the drop-down. Press execute to produce the result. As above, the box and whisker plot shows the substantially elevated gene expression for ERB2 in the 3+ group. By selecting the summary view, we can see that there is a statistically significant result. This indicates that at least one of the groups is different from another of the groups. The box and whisker plot shows that it is highly likely the 3+ group is the differentially expressing group. We could easily verify this with further analysis, which we will save for a later lesson. This concludes our HER2 ERB2 expression demonstration. Thank you.